Hello to everyone. I'm Oscar Palomares, professor at Complutense University of Madrid, and it is my great pleasure to share with you our last results published in Allergy, Allergoid-mannan conjugates imprint tolerogenic features in human macrophages. Allergoids conjugated to mannan are vaccines that are produced by immunotech by conjugating allergens to mannan purified from Saccharomyces cerevisiae. These allergoids conjugated to mannan target dendritic cells via C-type leptin receptor, enhancing allergen uptake and promoting tolerogenic dendritic cells able to polarize allergen-specific regulatory T cells. We have previously shown that aluminium impairs these tolerogenic properties by inhibiting mTOR and metabolic reprogramming in human dendritic cells. Monocytes are one of the first cells recruited into injection sites during subcutaneous immunotherapy, and they can differentiate it into dendritic cells or macrophages. We previously shown that allergoids conjugated to mannan reprogram monocytes into tolerogenic dendritic cells by mechanisms depending on epigenetic and metabolic reprogramming. In this study, we wanted to study how allergoids conjugated to mannan can polarize or condition differentiation between from monocytes to macrophages. So for that, we first isolated or differentiated macrophages from THP1 cells and differentiated them under pro-inflammatory M1, pro-allergic M2A, or protolerogenic M2C conditions, as well as in the presence of allergoid conjugated to mannan to assess and compare the induced phenotype. As you can see here, Allergoids conjugated to mannan promotes the generation of tolerogenic macrophages, as determined by the expression of IL-10 and by the lack of expression of other markers such as CCR7, CD80, CD206, or IL-6. So next, we isolated monocytes from healthy donors in humans and differentiated them under pro-inflammatory M1 conditions alone or in the presence of allergoid conjugated to mannan. As you can see here, the vaccines promoted the generation of tolerogenic macrophages here in blue, as you can see by the expression of CD163, CCL2, IL-10, and other pro-inflammatory protolerogenic markers. The same results were obtained under M2A pro-allergic differentiation. Supporting this data, allergoid conjugated to mannan generated macrophages that produces significantly higher levels of IL-10 and lower levels of pro-inflammatory cytokines TNF-alpha and IL-6 than pro-inflammatory macrophages. Finally, we wanted to study the capacity of these tolerogenic macrophages to polarize T-cell responses. And for that, we isolated monocytes from allergic individuals, differentiated into the different conditions of macrophages, and performed co-culture experiments under antigen-specific responses. So as you can see here, the macrophages generated by allergoid conjugated to mannan promoted significantly higher levels of allergen-specific FOXP3 positive regulatory T cells that produces significantly higher levels of IL-10 than those T cells generated by the pro-inflammatory macrophages. So in conclusion, our data clearly demonstrated that allergoids conjugated to mannan promotes the generation of tolerogenic macrophages, producing significantly higher levels of IL-10 and lower pro-inflammatory cytokines such as IL-6 or TNF-alpha, and in addition, they promote the generation of allergen-specific FOXP3 positive regulatory T cells. This data contributes to enhance our knowledge on how these next generation vaccines for allergen-specific immunotherapy could promote healthy immune responses against allergens. And that is all I wanted to share with you today. Thank you very much for your attention.